Uh, Dear students, the topic of discussion for today is biological molecule. This is our new chapter, biological molecule. Okay. Biological molecules are those molecules that form our living cell. Those molecules that form our cell are those molecules that need for the function of our cell. Right? Those molecules that need for the structure and function of cell. Of living cell, right? Those molecules that need for the structure and function of living cell, right? Branch of biology that deal with the study of these molecules is called molecular biology. Branch of biology that deal with the study of these molecules called what? Molecular biology. In molecular biology, we studied these molecule, the structure of these molecule, how it is, okay? While branch of biology in which we are studying the reactions between these molecules and how it is utilizing in our body, that branch of biology is called what? Biochemistry. Actually, biochemistry, right? Actually, biochemistry deal with the reactions or the metabolic processes that happening in our cell, which involve these biological molecules, right? That is called biochemistry. Now, example of biological molecule that we are going to study now. Carbohydrate, carbohydrates, protein, lipids, nucleic acid. Right? Now, nucleic acid, we have two types. One is DNA, another we have RNA. Ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. If you want to write, so DNA deoxy ribonucleic acid, okay, while RNA ribonucleic acid. These molecules form our cell. these molecules need for the function of our cell. Now, all these molecules, commonly contain specific, like these molecules may contain carbon, and all these molecules carbon is present. And all these molecules hydrogen is present. Carbon, hydrogen is present in all these molecules. So that's why all these are what? Organic molecule. Because it contains carbon and hydrogen. Carbon, hydrogen containing molecules are organic. Okay. Now these two are present in all these molecules. While in other elements you can also find in some, right? Like for example, you can also find nitrogen in that. That is present in nitrogenous basis of DNA and RNA. You can also find sulfur. The sulfur is present in some amino acid of protein, right? And remember, carbon, hydrogen, with this I will also put it here, oxygen. Oxygen is also present in all of these molecules. Carbon, hydrogen, and oxygen, that is present in all these molecules. Okay. While nitrogen and sulfur you can also find in protein, and you can also have, and nucleic acid is, I have example of nitrogenous basis that is present in the DNA. Okay. All right. Now, all these molecules are actually macromolecules. All these are what? Macromolecules. Okay. What is macromolecule? Macromolecules are those molecules, right, whose size is large. So macromolecule, a molecule having large size. Right? So all these molecules, protein, nucleic acid, lipids, all these are what? Macromolecules. Here I will write polysaccharide. Polysaccharide. Polysaccharide is actually the type of carbohydrate, right? So that formed by the simple sugar, that formed by simple carbohydrate. That's why I included here polysaccharide, because here macromolecule should have a large size. So polysaccharide have large size. Okay. Now these are the example of macromolecule. Now how these molecules form? These molecules contain a basic unit, like a basic. These molecules form from small basic units. Okay. Okay. This is called what? Let us discuss. Let's suppose I will write you here. The first one I am taking example of carbohydrate. Okay. Carbohydrates having different types, but the one that is macromolecule and that having large size, that is called polysaccharide. That's called what? Polysaccharide. And polysaccharides form from what? From basic unit, that is small basic unit, repeated unit. That's called what? Monosaccharide. Right? Monosaccharide. Example of monosaccharide we have like glucose, fructose, galactose, right? They form what? Polysaccharide. They will form a large molecule carbohydrate, polysaccharide, right? For example, one example of polysaccharide is cellulose. This is example of polysaccharide. Example of polysaccharide, another I have starch. Cellulose present in plant cell wall. Am I clear? This cellulose is present in plant cell wall. Starch is the store form of glucose and plant. While glycogen is the store form of glucose and human or animals. Right? Now, if you study these polysaccharide, so the basic unit of these polysaccharide is what? Glucose. Several glucose, I will represent here glucose by G. Several glucose combined together repeatedly again and again, and they form what? Polysaccharide. Right? When same unit repeated again and again, let's suppose in this case glucose repeated again and again, so they make what? This glucose is the basic unit in it, and that make polysaccharide. Am I clear? No. Now, if you check it here, this glucose is the smallest unit here, right? The small unit that's repeated again and again, and that form a large molecule, a long chain. So in this case, you know, glucose is actually monomer. Monomer is what? A repeated unit. What is monomer? Monomer is a simple molecule which is used as a building block for the synthesis of a polymer. So monomer is actually a simple molecule which is used as a basic building block for synthesis of what? Of a polymer. Now question is, what is polymer? Right? Polymer is actually a large molecule. Polymer is actually a large molecule. Once again, listen me, simple molecule which is used as a basic building block for synthesis of polymer. Okay? Am I clear? So this is polymer. Now question is, what is polymer? So polymer is actually a large molecule made from many similar repeated units. Polymer is what? A large molecule Polymer is actually a large molecule made of made of many similar repeating subunits. Repeating subunits, right? Joined together in a chain or whatever, just keep it in mind that large molecule made of many simple repeated subunits. Now in this case, in case of carbohydrate polysaccharide, in this case I give you example of cellulose or starch. Both made from what? Glucose. So glucose repeated again and again, and they form a huge molecule called what? Polysaccharide. So in this case, this glucose is monomer. Monomer, right? Here make it correction, monomer. Okay, monomer, the small unit. Now, this monomer repeated and they form what? Polymer. This is actually what this complete chain is, polymer, while glucose, one glucose is what? Monomer. So, several monomers combine and they will form what? Polymer. In this case, example is what? Polysaccharide. Now, if we take the example of protein, but remember boys, one thing you should keep it in mind. For example, this is one monomer, right? And this is another monomer. Okay? When these two monomers are joining together, right? When they are joining together, definitely they will join together and they will form what? A large molecule called polymer. So when they are joining together, this reaction is called what? Condensation. The reaction in which two monomers join together is called what? Condensation. In condensation, what will happen? When both react, water will eliminate. Usually it is joining through condensation. The reaction in which two molecules join together chemically with the release of water is called condensation. While let's suppose if I'm converting a polymer to its monomer, this is a polymer. And I'm going to convert it to the small basic molecule. So at that time, I have to add water. For what? For breaking up this bond. So addition of water for the breakdown of a chemical bond for the breakdown of a chemical bond, it's called hydrolysis. Am I clear? This is called what? Hydrolysis. Right? These monomer will separate from each other. Addition of water for the breakdown of chemical bond is called what? Hydrolysis. It means that if I am converting monomer into polymer that reaction is called condensation. While if I'm converting I'm converting polymer into its monomer, so that is hydrolysis. Am I clear? All right. Now let's come toward protein. You know, basic unit of protein is what? Amino acid. Amino acid. About 20 amino acids that need for our body proteins. Now these, for example, this is one amino acid, okay? This is another amino acid. This is another amino acid. This is another amino, like this. Now these amino acids are actually the repeated unit of protein. So in this case, for protein, these are monomer. These amino acids will join with each other through a bond called what? Peptide linkage. This bond is called peptide linkage. For when this bond is going to form, so at that time, what will happen? Water will eliminate. Hydrolysis will take place. Sorry, condensation. Am I clear? So H mean water molecule eliminate one here, one here, one here. It depends on number of bonds. Okay. So in this case, monomers are what? Amino acid. While polymer, we have protein. Okay. Now come to our nucleic acid. We have two types of nucleic acid. One we have DNA, another we have RNA, ribonucleic acid. Okay. Today the topic of discussion we have carbohydrate. Okay. Carbohydrates are actually organic molecules that are needed for our body. You know, it's actually, you know, carbohydrate having very important role in our body. The first thing is, you know, glucose is actually carbohydrate that provide us what? This provide us energy during respiration. Okay. Second, you know, carbohydrate also play important role in structure formation, like you know, cellulose is it's a carbohydrate. It's the component of plant cell wall. Is it okay? Now, carbohydrate can also play important role in bacteria. It's also
play important role in bacteria-cell wall formation, right? Like, bacteria-cell wall consists of what? Peptidoglycan or peptidoglycan. This glycan is actually carbohydrate part of that. Is it clear? And there is also different types of glycoprotein in our cell membrane. Okay, so it means carbohydrate also play important role in our structure formation, right? In different parts of the cell. Okay, is it clear? Now, carbohydrates contain three essential elements, right? Carbon, hydrogen, carbon, and oxygen. These three elements combine and form what? Carbohydrates. Is it clear? We have three types of carbohydrates. Three types of carbohydrates. Okay, one thing important rule of carbohydrate is, carbohydrate also in our DNA, right? There is what? Ribose sugar. In our DNA, deoxyribose sugar. So this is also carbohydrate, okay? And our RNA also we have what? Oxyribose or simply ribose, not ribose sugars. Okay, that one is deoxyribose and this one is ribose sugar. That's present in our DNA and RNA. It means in the formation of nucleic acid, also carbohydrate play important role. Okay, that carbohydrate is the part of that nucleic acid. There are three types of carbohydrates. The first one we have monosaccharides. The first one we have monosaccharide. Second one we have disaccharide. Disaccharide, right? And the third one we have polysaccharide. Disaccharide, and third one we have polysaccharide. Polysaccharide. So monosaccharide, disaccharide, and polysaccharide. Now the first one we will discuss about monosaccharide. Actually, monosaccharide is sample sugar. Monosaccharide this is sample sugar. This monosaccharide is actually the basic unit for the formation of disaccharide and polysaccharide. This monosaccharide is the basic unit for what? For disaccharide and polysaccharide. It is simple sugar. Simple sugar means it can't be hydrolyzed further. I will clear you this point when I start disaccharide. Can't be hydrolyzed further. Further into simple. It can't be, it means it's the basic unit. You can't, if you add water to that, it can't be converted into more simple form. So this is the simple form of carbohydrate. Okay. Another, you know, this is more soluble as compared to other carbohydrate. It is soluble in water and sweat and test. Okay. Example of monosaccharide, we have glucose. These are the example of monosaccharide. Glucose, fructose, and galactose. Okay, these are the example of monosaccharide. Now, you know, monosaccharide further divided into different types on the basis of carbon, on the basis of number of carbon, number of carbon atom in that. Right? For example, if a monosaccharide having 3 carbon, right? So that is called triose. If a monosaccharide having 4 carbon, it's called tetrose. If a monosaccharide having 5 carbon, that is called what? Pentose. If a monosaccharide having 6 carbon, so that is called hexose. Right? So these examples that I write to you here, the formula for all these is what? C6H12O6. It means in this molecule we having 6 carbon atom. This is 6 carbon atom. So this is the example of what? This is the example of hexose. Why? Because these contain what? Only a 6 carbon atom. The chemical formula for glucose, fructose, and galactose same, but the structure is different. And we know already in chemistry, a molecule having same chemical formula, but they are structurally different from each other. So that are called what? Isomer. So it means these 3 monosaccharides are isomer of each other. Okay, in which monosaccharide these 3 are? These belong to hexose category. Hexose means they contain 5 carbon atoms in their structure, while pentose contain 5 carbon atom. An example of pentose, I will give you ribose sugar in your RNA, or deoxyribose sugar in your DNA. That is 5 carbon molecule. Am I clear? All right. So the general formula for this is, you know, the general formula for this monosaccharide, we have CH2O, right? Here I will put it here, Y. What does it mean? It means that Y can represent you like, you know, for example, if I put it here, for example, glucose, right? So for the glucose, this Y I will put it here 6, right? Remember, what does it mean? It means that this formula means that the ratio between mostly this is applicable. Hydrogen and oxygen should be what? 2:1. Means the ratio between hydrogen and oxygen is 2:1, just like water. Just like what? Water. Right? So that's why carbohydrates also called hydrated carbon. Means the ratio between in all carbohydrates, you know, this ratio is, I'm talking about monosaccharide. This ratio is 2:1. Okay. Now, you know, if I put it here 6 for the glucose, so 6 multiply 1, so carbon 6. 6 multiply 2, 12. And 6 multiply 1. So this is formula for the glucose. I will apply this formula to others also, you will check. Is it clear up to here? If you want to take hints, you can. All right, boys, look at here, types of sugar, molecular formula and example, right? So molecular formula, the first one I will write for, you know, glycerol aldehyde. Glycerol aldehyde. Glycerol aldehyde. Glycerol dehyde. Glycerol aldehyde. Okay. All right. Glycerol aldehyde. Another one molecule I want to write it here. Threose. These are the name of carbohydrate that I am writing. Ribose and glucose. Okay. If you check here, C3 glycerol aldehyde formula, we will write it here CH CH, okay, O3, right? And another right for all CHO, right? And here, write it here, CHO. Okay. Now what we have to do here, this one is glycerol aldehyde is the example of what? Triose. This is example of triose. So this we will put it here 3 here. This triose is the example of what? Tetrose. So here you will put it here 4, and here you will put, this is the example of pentose 5, right? And here you will put it here what? 6, because this glucose is example of hexose, 6 carbon molecule. Correct? Now up to here it's correct. Now according to my formula, that the ratio between hydrogen and oxygen should be what? It should be 2:1. It should be 2:1. So what we have to do, if this 3 carbon, we have 3 carbon, right? So 3 carbon, so it's here should be what? 3. Okay. So 3 multiply 2, 6. 4 carbon, so here should be also 4. So 4 multiply 2, there 3 multiply 2, 6. Here 4 multiply 2, 8. And here 5 carbon, so that, you know, according to our formula, it is like formula was CH2O, right? So this Y should be what? It depends, like for example, I put it here for this molecule 3. So definitely that 3 will multiply with that 1 of carbon. So 3 multiply 1. So for that, we also have CH2O, right? For pentose. Here when I put it here 5, so 5 multiply 1, so 5 carbon. 5 multiply 1, 5 oxygen, right? So 5 multiply 1, 5 oxygen, and 5 multiply 2, 10 hydrogen. Okay, for this also same way. 6 multiply 1, 6, right? And 6 multiply 2, because the ratio should be 2:1. So 6 multiply 2, 12. Am I clear? So which type of carbon we have? This one, this is triose. Which one this is? Which type of carbohydrate this is? Tetrose, right? And which one this is? This is pentose. And which one this is? This one we have hexose. Am I clear? So these are what types of monosaccharide. So if you want to take hints, you can take. All right, boys. So up to here we discuss about monosaccharide, the simple sugar, right? And that cannot be hydrolyzed further. And then we discuss about the general formula for the monosaccharide, right? This formula, and then we discuss about what triose, pentose, hexose, right? All names of all sugar into with oath, glucose, fructose, and galactose, and two common pentose. We have ribose and deoxyribose, right? Up to here we discussed. This slide we already did. All right, now molecular and structural formula for glucose. Okay, glucose is 6 carbon molecule, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Is it okay? All right, so this is the structure of glucose, right? You can see here, this is approximate, both are same, but here they just wrote it what? CH2OH. CH2OH. Structure formula of glucose OH is known as hydroxyl group. There are five in glucose, right? So this OH is what? Hydroxyl group. This is what? Hydroxyl group in glucose. Okay, and this one you can see this is just a splitted form of glucose, and this one they just wrote it this one in CH2OH. Okay, so it means both are same structure. Okay. Is it clear? Now, this glucose, you can find it in the ring form. When it is in the solution, that is more stable form, you will find it in the ring form. Okay. One important aspect of the structure of pentose and hexose is that change of glucose atom is long enough to close up on itself. They can close it on itself. You can check. You can check here. It's converting into what? To ring form in a solution. Check it here. You can see here, this ring form is now, this stretch form is converting into ring form. Now, for the ring form, the important one for us is you can check here, boys. You know, ring form, this is a straight form of glucose, right? So this ring form will form between what? Between carbon, this carbon number 1 and carbon number 5, right? So they will form bond. Is it clear? You know, the hydroxyl group with carbon number 5 and right with carbon number 1. Is it okay? So the mechanism is not in our syllabus, so we will not do the mechanism, but which one is in our syllabus, we will do that. Okay, so this O, right? O of carbon number 5 and carbon number 1, right? So both will form what? They will make a chemical bond, and that will convert into ring form. So one we have alpha glucose, another we have beta glucose. Is it okay? So one we have alpha glucose and another we have beta glucose. So I don't want to go to the mechanism that how this bond form, right? How this oxygen bridge form, and no need of that for you people. Only you have to know about You have to know about what you, if you want to do, just for time being, just keep it in your mind. For example, for example, what will happen with this? For example, this negative and this positive, right, in this OH group, but no need of that mechanism for you. So I don't want to discuss that mechanism. Okay, just keep it in mind. This carbon number 1 and carbon number 5 will join and they will form a ring form. And ring form, you have to know about, two ring form we have. One is alpha glucose, another we have beta glucose. Is it okay? And alpha glucose, this depend on this carbon number 1. If this hydroxyl group is down from carbon number 1, so that will be alpha glucose. If the hydroxyl group is up from carbon number 1, that will be beta glucose. Is it okay? So this is important point that you have to know about that. Dear students, in today's lecture, we will talk about ring forms of glucose. This is glucose, and the structure of glucose is straight, right linear. But, in more stable form, when it is in solution, so it is in ring form. Okay, so we have two ring, you can check here, this straight form will convert into ring form by formation of bonds between, by the formation of bonds between this carbon number, okay, this oxygen of carbon number 5, okay, and this oxygen of carbon number 5 and carbon of, and with carbon number 2, right? So they will form bonds, but we no need for us to know the mechanism, okay? And now when these two form bond, right, so what will happen? This glucose will convert into ring form. You can check here. Now the important thing for us is this carbon number 1, right? You can see this is carbon number 1. If the hydroxyl group is down from carbon number 1 downward, so that we have two, remember, we have two ring form of glucose. One is alpha glucose, another is beta. Now it is on the basis of hydroxyl group for carbon number 1 of ring form of glucose, right? If this hydroxyl group is downward, so we call that alpha glucose, okay, from carbon number 1. Now if this hydroxyl group is above carbon number 1, we call that beta glucose. Am I clear? So we have two ring form. One is alpha, okay, and another one is beta, right? One form we have alpha glucose, okay. Second one we have beta glucose. Now in alpha, OH group, hydroxyl group is below carbon number 1. Below or downward from carbon number 1, while hydrogen is above, right? While in beta glucose, hydroxyl group will be above. Am I clear? So this is about two ring form of glucose. Now, what is the rule of these monosaccharides? Up to here we discussed about different monosaccharides. Now we have to know about rules of monosaccharide in our body, right? Or in living organism. Okay, first one, glucose need for what? For respiration. In our body to provide us energy. Glucose provide us what? Energy. Energy through respiration. Am I clear? So it is the store form of energy, short-term store form of energy. While this glucose can also be stored in our body for a long time, like in form of glycogen. Several glucose will come together and they will form what? Glycogen. So in our body, it can be stored for a long time. Right? Whenever our body needs energy, this glycogen will convert back into glucose and respiration of that glucose will happen and will give us energy. So in animal, it is in glycogen form or in human. Right? While this glucose can also be stored in form of starch and plant. Okay. Now, another monosaccharide, we have ribose. Right? Ribose is present in our RNA, ribonucleic acid. Right? RNA consists of basic unit called what? Nucleotide. RNA consists of basic unit called nucleotide. So several nucleotides will form what? RNA. This ribose sugar is component of what? Nucleotide. This ribose sugar is component of what? Nucleotide. Now, ribose is actually 5-carbon sugar pentose. It is monosaccharide. Another we have deoxyribose. This is also ribose sugar, but this is deoxyribose. Deoxyribose means, there is, instead of, there is some structural difference about ribose and deoxyribose. For example, this is a ribose sugar, okay? You can check this is a ribose sugar, for example. Normally in position number, carbon number 2, this is carbon number 2. Am I clear? So this is carbon number 2. Normally at that carbon number 2, it is hydroxyl and hydrogen in this ribose sugar. But in deoxyribose sugar, this hydroxyl group is replaced by hydrogen. That's why it is called deoxy, means there is no oxygen at carbon number 2. Like this with carbon number 2. This instead of hydroxyl group, there is what? Hydrogen. It means hydroxyl group replaced by hydrogen. That's why this is called deoxyribose. One oxygen is missing here. This oxygen is not there. Instead of that, there we have Instead of that, we have hydrogen. That's why this is called deoxyribose. But still it is monosaccharide. And this deoxyribose is present in, DNA consists of small unit called what's nucleotide. So this deoxyribose sugar is present. It is the part of nucleotide of DNA, right? So deoxyribose is present in our DNA. It means that this ribose sugar can form our DNA and RNA, and DNA RNA collectively called nucleic acid. Am I clear up to here? So this is the important role of monosaccharide in our body. Now we are moving toward disaccharide. Now we are moving toward disaccharides. Disaccharides is the name indicates di for two. Saccharin from sugar for sugar. So it means this disaccharides contain two sugar. How two monosaccharides? For example, this is one monosaccharide and this is another monosaccharide. Both will combine and that will form what? Disaccharide. One monosaccharide and another monosaccharide, both will combine through a bond. That bond is called glycosidic bond. Through glycosidic bonds, two monosaccharides will combine together and they will form what? Disaccharide. Is it clear? As compared to monosaccharide, this is less soluble and it means less sweet as compared to monosaccharide. Now, it means that if I do hydrolysis of this disaccharide, so it will give me what? Hydrolysis of this disaccharide, it will give me two subunits or two monosaccharides. It will give me two monosaccharides, right? How? For example, if I add water to this disaccharide, so the through water, right, by utilization of water, breakage of this bond will take place. Water plus enzyme, that will break that bond and will give me what? Two monosaccharides. Two monosaccharides. So disaccharides are those sugar that give us two monosaccharides through hydrolysis, through addition of water. If you remember, I told you in the topic of monosaccharide that it will not further hydrolyze. Why? Because if I'm adding here to water to this glucose, there is no glycosidic bond. It will not break further. That is the simple form of carbohydrate. While here, disaccharide on hydrolysis, it will give me two monosaccharides. Right? Example of disaccharide, we have sucrose, okay, maltose, and lactose. Am I clear? In all these three, what is present? In all these three, we having glucose. Glucose, glucose, okay, and glucose. In this sucrose, we have another monosaccharide that is called what? Fructose. That is called fructose. Okay, this maltose having what? Glucose. Am I clear? And this lactose having galactose. Dear students, in the last lecture we discussed about glycosidic linkage formation between two monosaccharides to form what? To form disaccharides, in which we discuss about example of sucrose. So you can check here how the condensation reaction will happen, okay, and how that water will remove. This mechanism is very important. If you see here, we have here alpha glucose, right? So that glucose form bond with what? Fructose in case of sucrose. Am I clear? Now, how this bond formation will take place? This hydroxyl group and plus hydrogen, right? So definitely this will positive and this will negative charge, oxygen having negative charge. So this water will eliminate. Now the reaction in which joining of two molecule take place and elimination of what water take place, that is called what? Condensation reaction. So in this case, if you see here, oxygen bridge form. Why? You can check when this water eliminates, so this oxygen will remain between these two molecules. So this oxygen bridge is actually what? Glycosidic linkage. The bond between monosaccharide of carbohydrate is called what? Glycosidic linkage, in which two monosaccharides joined with each other. So this is glycosidic linkage. Now, if you see here, in this carbon number 1 of glucose and carbon number 2 of fructose involved, right? So we call that 1,2 glycosidic linkage. This is what? 1,2 glycosidic linkage. Now, this is the same structure. You can check here, boys. This is the same structure, right? This is fructose, but this fructose in, you can see the structure is in rotated form, right? It doesn't matter. Okay, so it is in rotated form, right? So you can check here, this is monosaccharide glucose, and this is monosaccharide fructose, beta fructose, and alpha glucose. They will form what? They will form alpha, right? Alpha is glucose, right? 1,2 alpha 1,2 glycosidic linkage. Or if you want to mention the beta position, so alpha 1 beta 2 glycosidic linkage, because it's this two position. We have what? Fructose beta. We have beta fructose. That's why you can mention that like this also. Okay, so this is the formation of glycosidic linkage between two monosaccharides. Now, Alpha glucose and beta fructose will form what? Sucrose. Sucrose, the sugar that you are eating, that is sucrose. Okay. Now, how maltose will form? Let's move toward that. In this case, if you see 2 alpha glucose, sorry, so 2 alpha glucose come together and form what? Form glycosidic linkage. Alpha glucose means we are giving for the glucose from position carbon number 1, hydroxyl group is down, that will be alpha glucose. Here position carbon number 1, hydroxyl group down, that is alpha glucose. So again condensation reaction take place, elimination of water. Am I clear? Elimination of water and that form what? Glycosidic linkage. This oxygen bridge is what? Glycosidic linkage. Now, for example, you want to break that back, this reaction is reversible. So if you want to break, it means if you want to convert this maltose into glucose and glucose, so you have to add water. And addition of water in a molecule to convert it to subunit of that molecule, we call that hydrolysis. Am I clear? And here again carbon number 1 and carbon number 4 involved, so we called it alpha 1,4 glycosidic linkage. Which linkage? Alpha 1,4 glycosidic linkage is present in it. All right, boys, so for this fructose we are counting it what? From position carbon number 2, right? So the OH group is up, right? You can see because this fructose is in rotated form, so this OH group is above the carbon number 2, so we count it what? Beta fructose and alpha glucose, both will form glycosidic linkage. Okay, now let's move towards glycosidic bonds are covalent chemical bond that hold together glycoside, the chemical bond that hold 2 monosaccharide together. Okay, a glycoside is simply ring-shaped sugar molecule. Here ring-shaped sugar molecule are monosaccharide, right? So the sugar ring may be either 5-member or 6-members ring. So it may be either ribose, right, 5-member, and like pentose, or that may be hexose. Glycosidic bond formed by a condensation reaction, which means that 1 water molecule is produced during formation of glycosides. So the reverse reaction, the breakdown of glycosidic linkage, how you will do reverse? You will hydrolyze that, you will add water to that. Okay, we already discussed this. The oxygen bridge is called what? That is glycosidic bond. Already we discussed this, right? If you want to do it like, if you want to convert that disaccharide back to monosaccharide, you have to add water. This you have to know it, okay? When you are adding water, so from both sides, you will keep what? OH and OH. So both monosaccharide are now what? Separated from each other. And this reaction is called what? Hydrolysis. Addition of water to molecule to convert that into subunit, or what are the submolecules in that. Like here in this case, we have glucose and fructose. In case of sucrose, when we are doing hydrolysis of sucrose, we will get alpha glucose plus beta fructose. Am I clear? All right. A monosaccharide is a molecule consists, this is just summary of the previous lectures. A monosaccharide is a molecule consists of one single sugar unit with general formula this. A disaccharide is a sugar molecule consists of two monosaccharide joined together by glycosidic bond. A polysaccharide is a polymer whose subunit are monosaccharide joined together by glycosidic bonds. Many monosaccharides will come together in that will form what? Polysaccharide. If that are two, that will be disaccharide. Now, before moving toward polysaccharide, let's study about the reducing sugar. Reducing sugar include all monosaccharides such as glucose and some disaccharides such as maltose. The only common non-reducing sugar that we have to know about that it is sucrose, because most of our practical work will be related with this sucrose. So keep it in mind, reducing sugar we have glucose and maltose. Non-reducing we have sucrose. Now question is, what is reducing sugar? Reducing sugar having the ability to do what? Reduction. Reducing sugar are so called because they can carry out type of chemical reaction called what? Reduction. What means by reduction? It is the word, chemistry word. Reduction means, for example, I have A molecule and another I have B. This A gives electron to B. Am I clear? So what happened in this case? This B become reduced, right? While this A become what? Oxidized. Because A gives electron to B. So reduction reaction happen, electron gained by whom? By B. So B become what? Reduced, giving up electron reduction. While this A become what? Reduced. Am I clear? So in this process, you know, sugar will give electron. So that sugar will become what? Oxidized. To what it will give electron? Let's see. This is made use of in Benedict test using Benedict reagent. We are doing this test by using Benedict reagent. Benedict reagent is actually copper sulfate in alkaline solution and having distinctive blue color. It is in blue color. What is in blue color? Benedict solution. Right? Let me repeat once again. We have two types of sugars. One reducing sugar, another is non-reducing sugar. Reducing sugar having ability to do reduction, which means that they will give electron to another. So another will reduce, another will accept electron, while this will remain oxidized because it will give electron to another. So how we will check that the sugar is reducing or non-reduced? We will do for that Benedict test. In Benedict test, we are using Benedict reagent. And Benedict reagent is actually copper sulfate. Now, reducing sugar actually reduce soluble blue copper sulfate. What it is doing, it is actually this reducing sugar is reducing copper sulfate ion to insoluble black red copper oxide containing copper. The copper oxide will, when reducing sugar, okay, containing to insoluble black red copper sulfide. When this, when this dye receive electron, when this Benedict reagent receive electron, so that will convert into what? Black red copper sulfide. The color will become what? Black red. Is it clear? Now, reducing sugar, how it is doing, you can check here in Benedict region we having copper sulfate, right? So here this having what? 2 valency, right? Now, when it accepts electron from this reducing sugar, am I clear? When it accepts electron from this reducing sugar, so what will happen? This reducing sugar lost electron. When reducing sugar lost electron, so it means reducing sugar become oxidized. As I give you example of A and B. Now, this one, 2 plus, having this valency, need 2 electron, but here it gets 1 electron from reducing sugar. So your valency become only plus. Right? Now this copper in is in the form of what? Reduced form. Because this copper receive electron. Reduce means to receive electron from another. So now this become reduced. And reduced form, the color of this dye or copper sulfide will be red, brown or brick red. Am I clear? So how I will figure out that the sugar that is in the solution form in a tube with me, it is what? It is reducing or non-reducing. For that, I will mix that sugar with this Benedict reagent. If the color changing from blue to red, yellow, or like that, I will show you the range now. What are the range for that? So that will show me that how much reducing sugar is in this solution. This is the procedure, boys. Add Benedict reagent to solution you are testing and heat it in the water bath. Heat is compulsory here, boys. First, add Benedict reagent to that solution that you want to test. If reducing sugar is present, so the solution will gradually turn to green, yellow, orange to red, brown. If sugar is more, large amount, so the color will be red. Am I clear? So for this reaction, we need heat. That's why we keep that in water bath. If sugar in that solution is moderate, so that will be orange red. If there are traces of reducing sugar, that will be green, yellow. Green or yellow, okay? Or greenish yellow. If none, so if there is no reducing sugar, it will remain blue. Let's suppose if in my hands I have one solution of glucose, another solution of sucrose. Into both I put Benedict reagent. So what will happen? The sucrose will remain what? Blue, because sucrose is not reducing sugar. While glucose will convert into brick red. Why? Because glucose is reducing sugar and that can reduce Benedict reagent. That can reduce copper that is present in Benedict reagent. Is it clear? All right. So when you do, generally, this is just for your information, that how we will know, how we will compare our experiment. So the first thing is that you can also make standard before performing the experiment. That you have known quantity of glucose in tubes and you did already for that Benedict test. So the sample in which you want to check concentration of glucose, when you perform that, you will compare with that standards, that to which this sample in which I am going to check glucose or reducing sugar, to which it show resemblance. Before that, I should have a known, like, a tube containing known quantity of glucose. then it is, I will use that just like a standard. Right? For example, like in this, I put, for example, in this 10 of gram of glucose, and this 20, and this 30 gram, right? And when I take a sample of anything, fruit sample or anything, and I do Benedict test for that, and I figure out that the color is, means like, what is the concentration of that glucose in there. So I will compare that color with any standard. For example, that show resemblance with this yellow, so, or orange, sorry, orange. So it's like, you know, orange or yellow. Okay, so I will reach to a point that there are what, if it is orange, so a bit more than it is moderate. But if it is yellow, so it means there are traces of reducing sugar. Okay, and I will know that maybe my in this the concentration is approximately 20 gram, because my standard contain 20 gram or near about that. Or you can also use a colorimeter, right? You can also measure the time taken for the color change through, if it take less time, it means reducing sugar or more. One is that. Another, you can also use colorimeter. You know, usually we are facing problem in color judgment. So in order to improve your experiment, you have to use for this color judgment better way to do it by colorimeter. It's a device that show you, the color intensity, that the color is less or more. So you can use that method. That is the best method to check such experiment in which you are comparing colors. Am I clear? All right. So another way, another one we have, now you can check here with 0.1% glucose, right? With distilled water is control, it's supposed to remain same. This I take it is a control. And with sucrose solution, so with sucrose solution, right? So sucrose solution, I know that sucrose solution will not give me a result, right? Because that is non-reducing sugar. So that also remain blue, and this remain what? Also remain blue, because this is water, it will also not give me result. So these two, right? While the one that having with 0.1% glucose solution, so the color is little bit near to blue, and it is color green and blue like that it's supposed to be. And this one is orange with the less, and right, and this is little bit brown near to yellow, yellow more than yellow, right? So it means this contain 10% glucose, and this color didn't change because sucrose solution is not reducing sugar. So sucrose solution can't reduce the Benedict dye. So because of that, this color remain blue, right? All right. Now, some disaccharides such as sucrose are non-reducing sugar, and it will get a negative result from Benedict test. So it means what is non-reducing sugar? Sugar that give me negative result with Benedict solution. Test that give me negative result, the sugar that give me negative result that didn't change from blue to other color, so that will be non-reducing sugar. Okay. All right. If both are reducing and non-reducing sugar are present, so the precipitate obtained in the test will be heavier than the obtained in Benedict test, right? If there are both, you know, if there are both in a solution, right? So the precipitate, you know, like the thing that is inside there, so that precipitate will be more heavy as compared to the one there is only like, you know, is compared to that you obtain in Benedict test. Obtain in Benedict test means like, you know, if you, for example, let's suppose I have one glucose, right? For example, glucose here is 10 gram, right? And another tube I have 30 grams of glucose and plus sucrose also 20 gram, right? So this both color will be same, right? Right? Why? Because 20-30 grams glucose and 30 gram glucose, both same glucose, right? So definitely this glucose will give me reducing positive, but then it will be a bit difficult for you to know that there is also something other. Then you will figure out that this one tube is heavy, right? So it means there is some precipitate of non-reducing sugar or something other. Okay. Now, let's suppose if I'm going to do test for non-reducing sugar. So it will give me test what? It will give me negative with Benedict solution. But still I want to confirm it is, it means that exactly it is non-reducing or no. So what I will do, in non-reducing sugar test, the disaccharide broken into monosaccharide. First of all, we will broke, for example, I have sucrose, so sucrose contain what? Fructose plus glucose, right? So definitely, if this contains this glycosidic linkage and I mixed it with Benedict solution, the test will negative. But still I want to check it. So for this non-reducing test, what I will do, I have to, for example, I do this test, I know that there are sucrose and I do Benedict, it is negative. Let me check how I will confirm it that this is non-reducing. I will add chemical hydrochloric acid. I will add any acidic like hydrochloric acid or sulfuric acid, right? When you add in this solution, so what will happen? The chemical reaction is hydrolysis and can be brought about by hydrochloric acid. So this hydrochloric acid in the presence of water, what will happen? It will do hydrolysis. Okay. Bond breakage, it means simply just keep it in mind. A chemical reaction is hydrolysis and can be brought about by hydrochloric acid. Simply just keep it in mind. This glycosidic linkage breakage will happen with the help of acid, HCl. Right? So when you break this bond, so you will get two monosaccharides, one mono and another monosaccharide glucose. Now these monosaccharide are what with me? The constituent monosaccharide will be now, these are now reduced, these are reducing sugar. And their presence can be tested by using Benedict test after the acid is being neutralized. Now remember boys, one thing here is very important. That let's suppose I have sucrose solution in my hand, in this tube, right? Now what I did first, I will put acid to break glycosidic bond. For the breakage of acid, I put glycosidic. Now this solution now contain what? That monosaccharide, because this hydrochloric acid break that bond. Right? Now, if I am putting Benedict solution directly to it, so this acid, Benedict solution is not working in acidic environment. So that will not give me result. So before adding Benedict solution in this, I will put some alkaline thing, so that I neutralize the acid. And when you are putting that alkaline in that, you will see bubble production will happen. Bubble will form because you are neutralizing the acid. Am I clear? After that, after neutralization of acid, you will put what? You will put Benedict solution. Okay? They heat the sugar solution with the hydrochloric acid. This will release pre-monosaccharide and Benedict region needs alkaline condition to work. So you need to neutralize the test solution by adding alkalis such as sodium hydroxide. First, before adding, like this is that solution, so you put acid in that, okay, to do reaction, you will heat that, right? So when you heat that, it will remove monosaccharide. Then you will add what? Alkali, then you will add sodium hydroxide to neutralize that acid. And after that, you will add your Benedict region, and when heat it as you was doing for reducing sugar, and look for the color change. If the solution goes red now but didn't in the first stage of the test, there is no reducing sugar. If before the test it was not changing and now it become red, so it means that was what? That was non-reducing sugar. But if there is no still change in the color, then there is no sugar of any kind of sugar is not. If still no change, still the blue remained blue. So mean there was nothing regarding, there is no any sugar. This is something other. So let me repeat one time more. In my hand, I have what? A tube containing a solution. Tube containing solution, and when I put initially Benedict reagent, it give me what test? It give me the color didn't change, it remained blue, right? So this solution I took from a stock solution in the beaker. Now what I want to do, I want to confirm that is it sugar or no, what is it? So I will take sample in another tube now from that stock solution. And what I will do, I will put in that acid, so that acid break glycosidic linkage between and convert that into monosaccharide. Now, when I put acid, I will heat this. Why I will heat this? So that it increase reaction rate. When I heat this in water bath, what I will do after that, I will take this, right? And you know, it's heat in this stage. Even you can also like very gently, if you do through lamp, like a spirit lamp, very gently if you want to heat it, or in water bath, but the purpose is to provide heat to this, right? After that, what you will do here, you will still this solution contain acid, and if I'm putting directly Benedict solution, Benedict reagent will not work, because Benedict need alkaline condition. So I will not put directly that Benedict reagent. I will put in this what? Sodium hydroxide, and when I'm adding sodium hydroxide, so bubble will start producing here, and I will put up to that time this sodium hydroxide, that bubble, no more bubble produce, and no more bubble production will be the indication that there are no more acids. Once you neutralize your solution, put Benedict reagent. This is sodium hydroxide. Okay, once you neutralize it, put your Benedict solution. Put Benedict. And once you put Benedict, so again, what you should do, you should give heat in water bath and check for the color change. If the color change to yellow or red, like greenish, so it means that this one was sugar in this tube. In this tube there was sugar, and that sugar was non-reducing sugar. But if still color remained blue with you, is it was before the experiment, or it was in this tube. So it means in this tube we don't have sugar. In this stock solution we don't have sugar. Am I clear up to here? All right. So, dear students, today the topic of discussion is cellulose, right? So, cellulose is also polysaccharide. Basic unit of cellulose is glucose, right? Several glucose come together and that will form what? That will form cellulose. Now, the bond between are the glucose that involve in formation of cellulose that is beta glucose. Okay, cellulose is the most abundant organic molecule on the planet due to its presence in plant cell wall and its slow rate of breakdown in nature. Slow rate of breakdown in nature, it show more resistance, okay, for the breakdown through digestive enzymes. And another thing, it's the most abundant, plants are more in nature. And it's present in plant cell wall, so is the most abundant organic molecule on the planet. It is structural rule being mechanically strong. It's this having structural rule. This is mechanically strong, not like starch and glycogen. It is more strong and it gives support to plant. It gives uh, mechanical support to plant because it is present in plant cell wall. Beta glucose is present in this cellulose, and beta glucose, we know already that if carbon number 1 hydroxyl group is present above carbon number 1, so we call that beta glucose. So in cellulose we have beta glucose. Now let's move toward the structure of this cellulose. You can check here, cellulose consists of beta glucose and the bond between them is what? Beta 1,4 linkage. Cellulose is not branch. Remember, it is not branch. There is no 1,6 linkage. There is only beta 1,4 linkage. Check here, this is one glucose and this is another. So carbon number 1 of this glucose and carbon number 4 of this glucose both contribute in the formation of glycosidic linkage. Am I clear? Now, if you check here, this glycosidic linkage formed between two glucose, this is that glycosidic linkage. But if you see, one glucose is present like this, while the another one is rotated by what? By 180 angle. Check here. How? This is carbon number 1 of this glucose, this is oxygen. So this is straight, while this carbon number, this oxygen is down. So both are present opposite to each other. Am I clear? And the rotation of this beta glucose by 180 angle is play very important role to give a more strength to cellulose, and how we will discuss this now. Am I clear up to here? So you check here, this is one glucose, the another one is rotated. Check, you can see this oxygen. Oxygen is up here, it is down. Oxygen up here, it is down. So all these are what? These are glycosidic linkage between glucose molecule. So a long chain of glucose molecule forming beta 1,4 linkage that will form what? Cellulose. But several chains will come together. So let's see how they are doing. How that bond will form and how several chain will form cellulose. You can check here, in order to form glycosidic bond with carbon atom 4, where the OH hydroxyl group is below the ring, one glucose molecule must be upside down, rotated 180 related to another. So definitely the one is already rotated down, right? Now, the successive glucose units link at 180 angle to each other. Both are linking with each other by 180 angle, right? Now, this 180 is very important. How? The strength or hull, you can check here that, this oxygen can form hydrogen bond with another they can form hydrogen bond with hydrogen, or you can see here in this place this oxygen form hydrogen bond with this hydrogen, right? Hydrogen bond is the weak bond between what oxygen and high electronegative atom, like hydrogen, fluorine, and fluorine. The bond between, once again listen me, I think I should tell you again because I think I was wrong. The bond between hydrogen electronegativity is very less. Electronegativity is the ability of element to attract electron toward its center. So the bond between hydrogen and high electronegative element, fluorine, right? Fluorine and oxygen. These are high electronegative elements. So bond between these elements that that form between hydrogen and these elements, that bond is called what? Hydrogen bond. All right, boys. So please, boys, make this correction. This is not fluorine. This is nitrogen. So nitrogen, oxygen, and fluorine, the bond that form bond between these are high electronegative elements. They form bond with hydrogen, right? So that is weak bond, but still if many hydrogen bond come together, they are giving stability to a molecule. So from this rotation help us that this molecule can form many hydrogen bond. Like up they can form, you can check it here. This oxygen form bond with another glucose in the same strain, right? But this oxygen, you can check here this oxygen. This oxygen form hydrogen bond with another hydrogen up downstream, right? So all these strains are joined together with each other through what? Through hydrogen bond. And many hydrogen bond can give stability to a molecule. Actually, a hydrogen bond is a weak bond, but if there are many, so that are giving what? Stability. So that's why, you know, one glucose is rotated by 180 angle, so that there is more chances of hydrogen bond formation. It was in one plane, so the hydrogen bond formation will be only on one plane, right? But now if you rotate one glucose, so the bond from different sides, there is more chances of hydrogen bond formation of oxygen to more chances of oxygen to form hydrogen bond with oxygen. That's why it's rotated by 180 angle to give more stability to silicon. This is very important point by the point out. Now, between 60 and 70 cellulose molecule become tightly cross-linked to form what bundles called microfibrils. Several, about 60 to 70, like such change will come and they will cross-link through that hydrogen bond, right? So that will form what? Microfibril. Am I clear? Now, microfibrils are strung held together in bundles called fibril. Several microfibril. This is one microfibril. Like such, several microfibril will come together. Like this is another microfibril, right? And so several microfibril will again head with each other through hydrogen bond, and that will form what? Fiber. That will form what? Fiber. Am I clear? So that will form fiber, right? Microfibrils are in turn held together in bundles called what? Fibers by hydrogen bonding. Am I clear? So for example, this is one fiber and this will be another, you know, this is microtubule. So another microtubule. So that make what? Fiber. Am I clear? Now, several fiber will come together. They will be, means like they will form hydrogen bond again, and that will form what? Cellulose. So let me tell you once again. One chain of cellulose. So if 60 to 70 chain of cellulose, that will form what? Microfibril. Several microfibril will come together. They will form what? Fiber. And several fiber, this is one fiber, this is another, like count it one fiber, another. They will form what? Cellulose. Cellulose is extremely strong material, insoluble, tough, and durable, but slightly elastic. Cellulose fibers are straight and uncoiled. Am I clear? So a cell wall typically has several layers of fibers running in different directions to increase the strength. So it means several fibers will combine, right? And that will form cellulose, right? This is actually cellulose fiber, right? But several fiber come together, we call that cellulose, right? That fiber is also called cellulose fiber, right? But several fiber collectively we call that cellulose. Cellulose make up about 20 to 40% of average cell wall. Other molecules help to cross-link the cellulose fiber and some form what? Glue-like matrix around the fiber, which further increase the strength. You know, cellulose make up our cell wall how much? 80%, right? It's 40%. Other molecules also involved in the cross-linking. One in cross-linking, hydrogen bonding involved, right? But other molecules are also there, just like that help in the cross-linking to make bond between them, and also some molecule is just like a glue to in which these fibers are present to increase the strength of this. Cellulose fiber having a very high tensile strength and almost equal to the steel. Tensile strength, you know, right? If you are putting pressure over something, right? How much force you are applying force or something, and how much it can tolerate that force. The toleration of that force, like how they can tolerate. This called tensile strength, right? This is physics. So this means that if you put it both in, that will be very difficult to stretch or break. It's very high. And make it possible for a cell to withstand the large pressure that develop within it is as a result of osmosis. Am I clear? So this means that this sentence means be clear here. That this sentence means that cellulose fiber having more tensile strength, just like steel. We will say that it's just like steel. It can tolerate pressure. It's not like almost equal to, but we will modify here this sentence. Just like steel, it is very strong. Okay. Now, and make it possible for the stool. So if it is very strong, if cellulose is very strong, so it gives benefit to what? To the plants. For example, water moved to from higher or from dilute region to inside the cell more, and inside the cell more osmotic pressure develop. So this cellulose gives that strength to cell may not burst. If you keep that cell or plant cell in dilute solution, that will not burst easily as compared to animal cell. Why? Because that plant cell having cell wall, and that cell wall contain what? Cellulose, and that is more strong. Okay, that give mechanical strength to plant cell. Without cell wall, the cell will burst if it is in dilute solution. So these pressure help provide support for plant by making tissue rich and responsible for cell expansion during growth. This is also, responsible for cell expansion. So this pressure helps to provide support from the plant by making tissue rigid. It make the plant, if there is water pressure, so the plant cell will be rigid. And if it is more expanding, so that expansion also helps plant to cell to grow, right? Responsible for cell expansion. And if the cell is expanding, it means plant will grow, the cell will elongate. The arrangement of fiber around the cell helps to determine the shape of cell as it grows. Remember, cellulose fiber are freely permeable. As about cell membrane that is partially permeable, everything can pass through it. But in the gap of this cross-linking, in the gap of this cellulose through this, the water can pass through that easily. Is it clear? These are cellulose fibers. So it means cell wall is freely permeable. Then the control of ion flow, ion or water flow, that will control by cell membrane. All right, boys, if you check here, this is oxygen, right? So oxygen electronegative, hydrogen electronegativity is less as compared to this oxygen, right? So positive charge will develop on this side, partial positive, and this will be partial negative. I will tell you now what does it mean by this bonding. So this positive and negative can attract each other. So this is called what? Hydrogen bonding. Electronegativity of oxygen is more. Why? Because they can attract electron more as compared to hydrogen, right? So that's why I mean the hydrogen electron will be toward that oxygen, It's not like this oxygen give a complete electron to that, but it will be the hydrogen electron will be near to what? To oxygen. Am I clear? They will share electron, but that electron will be near toward oxygen. So this is called what? Hydrogen bonding. Now we will discuss this in detail. I hope here this is clear. So, all right, boys. So dipoles and hydrogen bonding. When atoms and molecules are held together by covalent bonds, they share electron with each other. When two elements share electron with each other to make the last shell complete and the molecule to make a stable molecule, that is called what? Covalent bond. Another bond we have in which a molecule lost electron, that is called ionic bond. Covalent bond, they are sharing electron. So let's suppose I have one hydrogen this and another hydrogen this, right? So hydrogen having one electron and one electron. First shell, we know that first shell contain how much electron total? First shell contain how much electron? 2 electron total, but hydrogen this one electron and this one. It means both are unstable. So what these two will do, they will come near to each other and they will share that electron. It means they will share. It means this two will count for this also, and this two will now these two will count from this also. So hydrogen, it will form hydrogen molecule H2. So this is now stable molecule. So this is called what? Covalent bond. Just in this case, if you see last shell, we need how much? 8 electron for this and for that also 8. So what happened? They shared one electron. They shared one electron from this and one from this. So they shared 2 electrons, right? So one from one side and one from another side. So they make their last bond stable. You can count 1, 2, 3, 4, 5, 6, 7, 8. Right or no? Once again, 1, 2, 3, 4, 5, 6, 7, 8. Correct? So 8 for this and this 2 can count for this also. See, 1, 2, 3, 4, 5, 6, 7, 8. It means the last shell 8 complete. By sharing one electron he share and one this element share, right? So by sharing electron, they both stable their outermost shell. This is called covalent bond. For example, in water molecule, two hydrogen atoms each share a pair of electrons with oxygen, forming a molecule with the formula H2O. In water molecule, what is happening? You know, the first thing, oxygen contain how many electrons, right? 1, 2. First shell complete for oxygen, right? But second shell contain how many electrons? 1, 2, 3, 4, 5, and 6 electrons. It means oxygen needs how much electron? 2 electron needs to complete his shell. 2 electron need by oxygen to complete his shell to become 8. While hydrogen contain how much electron? 1. And this hydrogen also contain 1. It means this hydrogen needs 1 electron, and it means this hydrogen needs 1 electron to complete his first shell, because first shell contains 2 electrons. So what will happen? This oxygen will share this electron with this hydrogen, and this oxygen will share this electron with that hydrogen. It means they form what? Covalent bond here. One covalent bond in another covalent bond. So hydrogen becomes what? Hydrogen share this electron with hydrogen, and hydrogen share his electron, right or no? And in this case, also oxygen share his electron and hydrogen share his electron with oxygen. So this bond that forms, this is covalent bond, and this is covalent bond. Hydrogen also becomes stable, 2 electron, and here hydrogen also becomes stable. And oxygen now also having 8 electron in last shell. 1, 2, 3, 4, 5, 6, 7, 8. Because the hydrogen electron will be count for oxygen now because they are sharing. So this molecule is now a stable molecule. This is H2O. Am I clear? Now, however, they form covalent bond, but the electrons are not shared absolutely equally. Why? Because, you know, in this case, oxygen electronegativity is very high. The attracting of electron towards its center for oxygen, that ability of oxygen is more, and hydrogen is very weak. The lowest electronegativity, what? Hydrogen, right? While electronegativity of oxygen is very high. So that oxygen is keeping that electron little bit near to himself. That's why it's not like this that it gives complete electron, hydrogen gives complete electron to oxygen. But no, he is little bit strong, so that's why he is more strong, not little bit. That's why it's attracting electron toward itself. It's near to oxygen. So partial, we call it partial negative. It's not complete give electron. If it give complete electron, then it will be negative. That will be ionic bond. But here, this is a coordinate covalent bond because it give electron is near to oxygen, for partially negative or partially positive develop on hydrogen. Why? Partially positive means the electron give toward the oxygen, it's near to oxygen, right? So the power, am I clear? Now, and electron having negative charge, that's why we are writing here negative, okay? And in this case also that oxygen is near to oxygen. This electron is near to oxygen. So it's also partial positive and partial positive. Now, those molecules in which there is charge, we call that what? Polar molecule. In which charge develop, that are called what? Polar. Am I clear? Now let's see. In water, the oxygen atom gives slightly more than its fair share, and so is a small negative. charge, written partially negative and partially positive. So this unequal distribution of charges is called what? Dipole. This is called what? Dipole. Now, in water, the negative charge oxygen of one molecule is attracted to a positive charge hydrogen of another. And this attraction is called what? Hydrogen bonding. Now, let's suppose, boys, if I have water molecule, be with me here, boys. Negative, positive, partial, negative. Am I clear? So another side, I have another water molecule, let's suppose. This is another water molecule, partial positive, partial negative, and partial, right, positive. So you check here, this oxygen can attract this partial positive. One water molecule can attract each other. This is actually hydrogen bonding. And I discussed with you already, hydrogen bonding is actually the bond between high electronegative atom element. Here, that element we have what? Oxygen. That is attracting what? Low electronegative atom, hydrogen. So the bond between hydrogen and high electronegative element. In this case, we have what? Oxygen. So this hydrogen bond, this is called what? Hydrogen bonding. This hydrogen bond is a weak bond. If several bonds come together, they will make this molecule strong. Am I clear? Now, this having very important rule now for our topic. You can check here, this is hydrogen bond between two water molecules. And these bonds are very important in structure and properties of carbohydrate and protein. Am I clear? This is hydrogen bonding. You can check dipoles occur in many different molecules, like oxygen, hydrogen, right? And carbon, oxygen, and nitrogen and hydrogen group, right? So between them, charge can develop because oxygen is high negative element, high electronegative. So negative will develop here, here positive, here negative, here positive, here positive, here negative, right? So these hydrogen bond can form between these groups because the negative charge part of one group is restricting the positive charge of another. Okay, now molecule which having group with dipoles, such sugar, are said to be polar. Those molecules such as sugars, right? Those molecules in which these charge can be developed that are called what? Polar molecule, right? And that molecule will be soluble in water. That are water lover molecule, and that water molecule is called hydrophilic molecule. Hydrophilic. It means polar molecule having charge and that are soluble in water. So such molecules are called what? Hydrophilic, water lover. If they have no charge, so that is called what? Non-polar molecule. They cannot dissolve in water. Why? Because there is no charge to make charge-to-charge interaction with water, ion-to-ion interaction. So that are called what? Non-polar. So non-polar are hydrophobic, that are water repellent, that are water hater. Such property make possible for formation of cell membrane. We will discuss the importance of this hydrophobic and hydrophilic nature in cell membrane. Am I clear up to here? All right.